So our second speaker is Kimberly Stegmaier from Harvard. Kimberly is a pediatric oncologist, hematology oncology particularly. She obtained her first degree from Duke and then her medical degree from Harvard Medical School and has stayed on there. She's now an associate professor of pediatrics and also a member of the Broad Cancer Program. So it's great to have her here today. And she's going to talk about genomic simplicity for a change, pediatric cancer and genomic simplicity. So is this here?
Thank you, Richard, and I want to thank Tyler and Mike for the invitation as well. And I have to confess, when Mike asked me to speak here today, I said, Mike, I don't really work on tumor heterogeneity. So I'm going to flip this a little bit on its head and talk about how we can actually learn from cancers that have relatively simple genomes. And so my laboratory works on a number of cancers that affect children largely. We use a number of different omic approaches, really with the goals of understanding the biology of these diseases, finding new drug targets, and ultimately new drugs. And so I thought I'd just provide a bit of a framework about pediatric cancer. So the most common cause of cancer in children, as you may know, is leukemia, particularly acute leukemia, with brain tumors now a close second, lymphoma third, and then a smattering of childhood solid tumors that follow. And so I always like to mention this, I think there's a bit of a misperception that, we're doing great treating childhood cancer, we're done, there's not that much more work to do. And, there is still over 15,000 children diagnosed each year with cancer in the United States. And cancer is still the leading cause of death by disease in children. So despite the fact that in comparison to adults, we're doing pretty well, this is always, I think, a startling comment to hear. And our survivors suffer a lot of long-term toxicity, and we're still learning about that toxicity as we study these children. But what I'd really like to focus on mostly today is this idea that pediatric cancers can really inform understanding of cancer more broadly, and that when you have a relatively simple genome, you can make observations that are more difficult to make in more heterogeneous, complex tumors. And I think I bring up this comment that, RB mutations were discovered in a very rare childhood tumor that involves the eye, and this really sparked the notion of the tumor suppressor. And so our survival rates are overall pretty good. Within each of these classes, though, there are diseases where the survival rates are quite poor, particularly for myeloid leukemia, as you see here on the bottom, and the metastatic subtypes of the pediatric solid tumors and some of the more high-grade brain tumors. But I think one question that one might consider is, are high survival rates in part related by simple genomes or limited tumor heterogeneity? And I think we have some cases where that may be yes, in some cases where that's clearly not so. And so with the next generation sequencing efforts, I think one of the major themes that emerged for us in pediatrics is that by and large, these genomes are really quite quiet. And I just highlight here a few pediatric cancers that I will mention over the course of the talk. One is rhabdoid tumors. These are generally defined by lesions in the switch-SNF complex with loss of SNF5. Ewing sarcoma, which is a solid tumor defined by EWS-ETS translocations, medulloblastoma, a pediatric brain tumor, and then neuroblastoma, which is a neural crest-derived tumor. And I'd like to mention another rare type of childhood leukemia called juvenile myelomonocytic leukemia, and in work that we've recently been focusing on with Mignon Lowe's laboratory at UCSF. These leukemias have an extremely low mutational burden, among, again, the lowest of those that we've sequenced. And I bring this up as one example of where I think a very important finding from our recent and as yet to be published work with Mignon, we've shown that in this leukemia, children who have a very low mutational burden have a much higher survival than those who have increasing numbers of genetic alterations. So I think this is one example where mutational burden does correlate with survival, and this has actually been shown in another independent unpublished study. In comparison to this tumor, rhabdoid tumors, where the survival rates are really miserable, and we really have made almost no progress in treating these children despite using very high intensity combination chemotherapy. And again, this is a tumor also extremely quiet genome, defined by SNP5 alterations. So I think you can see it's complicated. It's not a straightforward answer. But I think, though, that in this context, we can really use these tumors to make discoveries in this idea of the simple genomic background. So I think they can certainly inform basic mechanisms of oncogenesis that in studying a molecular lesion in a quiet genome, I think you can more easily learn about that lesion. I think that these tumors can inform us about biomarkers of response and synthetic lethal relationships engendered by molecular alterations. And I'd like to describe this a bit more to you today. And so I start with a published example from Charlie Roberts' laboratory that really sparked us to think about this more systematically. So Charlie's laboratory at the Dana-Farber studies rhabdoid tumors and switch SNP complex alterations. And so I think this is a lovely example where these tumors that have loss of SNP5 are quite dependent on EZH2, and this was shown in a genetic model. We in the community were very excited when this work was published by Epizyme that showed that it's not just a dependency on the total protein, but that enzymatic inhibition actually appears to be an interesting therapeutic concept for these patients. And in a recent early study, the one responder at the time of this information was a patient who had malignant rhabdoid tumors responding to an EZH2 inhibitor. So I think we are very excited about the prospects of using epigenetic therapy in these tumors. And I think this is going to become a theme in many of our difficult to treat childhood cancers that are defined by epigenetic lesions in simple genomes. So I think that we can also use these tumors to identify other biomarkers of response. And I was quite struck by these two publications, one that was led by the Broad Institute and the other by the Sanger and Mouse General, that systematically screened collections of small molecules against hundreds of cancer cell lines defined by molecular features. Most of the cell lines in these screens were from adult cancers, but in both of these papers, the two diseases that were highlighted were the same. It was a pediatric cancer, Ewing sarcoma. So in one paper, Ewing was identified as being highly sensitive to topoisomerase inhibitors. And this is in fact a therapy that we use to treat these patients. And in the second, Ewing was highlighted, and specifically EWS fly events were highlighted as engendering response to PARP inhibitors. There had also been another publication by Roel Chanayan's group suggesting that these compounds might be active in Tempus-Erg rearranged prostate cancer. And now these compounds are actively in clinical trials for children and adults with Ewing sarcoma. We had made a similar observation in our own work on neuroblastoma. And so in this publication, we did a screen with Jay Bradner's laboratory looking at response to BET bromodomain inhibitors in large collections of cancer cell lines. And the highest predictor of response to this compound class was amplification of MYCN in a pediatric solid tumor of neural crest origin called neuroblastoma. And so shown in red are the neuroblastoma lines, and in gray, the other cell lines in the screen. And so we found that both in vitro and in multiple mouse models in vivo, neuroblastoma was quite sensitive to inhibition with this molecule class. And in fact, this work was then extended by another group to medulloblastoma, also a disease of neural origin defined by both MYC or MYCN lesions. And that is a second disease, again, quite similar in many ways to neuroblastoma that's sensitive to these molecules. There's now a trial in development to test BET bromodomain inhibitors in children with these diseases. So I think this is a very nice example of where you can do a screen, you can do preclinical validation, and in a relatively rapid time, take the idea to the clinic. And so these pilot studies really have sparked a more systematic effort, collaboratively led by my laboratory, that of Charlie Roberts at Dana-Farber and Todd Golub at the Broad Institute, to take a more systematic approach to studying these cancers, and we've termed this the Pediatric Cancer Dependencies Project. So here, we're really taking this to scale. We're going to evaluate a number of different childhood cancers, really focusing on diseases that have subsets that are still quite poorly treated, define the molecular alterations quite deeply in these cancers, and I would put on that list now epigenetic features. Screen for vulnerabilities. We've done some screening already in shRNA. We're going to do now CRISPR screening, and we have some early data emerging, as well as drug and small molecule screening. Of course, this takes a fair amount of computational analysis, but really the ideas now are to match lineage with response to molecules or dependency or molecular features. And the hope is that if you find a relationship in an EWS fly rearranged ETS-driven cancer with a simple genome, that maybe that observation will extend to other more genomically complex ETS-driven cancers, such as TMPRSS positive prostate cancer. So my laboratory, in a series of pilot studies that preceded this much larger study, has focused on Ewing sarcoma, and I'd really like to spend the remainder of the time telling you about a couple of our Ewing projects in the laboratory and really show you where we're going. So this is the second most common tumor involving bone in children. About 250 patients are diagnosed each year with this disease in the United States. Children get treated with very intensive therapy. It's about a year of treatment. It includes 5 different cytotoxic drugs. We use what's called interval compressed chemotherapy, where every 2 weeks these children get this very intensive series of drugs. And then they get either surgery or radiation to the bulky site of their disease. And for these children, our curves actually have improved over the years, somewhere around 70 or 75%. But you can see the lower curve on that survival curve. Those are the children who present with metastatic disease. Their survival rates are somewhere around 20 to 40%, depending on the site of metastasis. And those curves have not moved at all in the last decades. And we really don't have any molecular features yet, although we may have one now, of who those children are or why those children have overt metastasis at diagnosis. But clearly they do poorly, and our current therapies aren't enough. Relapse is nearly always fatal for these children. And so what was known about EWS fly pre the genomic era, and then kind of what do we know now? So this is an ETS-driven tumor. EWS fly was discovered over 20 years ago. This takes the strong transcriptional activator, EWS, and fuses it to the DNA binding domain of fly, creating a fusion transcript. The cell of origin of this disease is uncertain, and we really have lacked the elegant models that Tyler just showed us. There's no reported faithful genetically engineered model of this disease, so it does limit our ability to study this disease. It's also been difficult to establish patient-derived xenograft models, so our tools are somewhat limited. So this is really data hot off the press. We had completed an shRNA screen, and we were very disappointed that fly-directed hairpins did not score. We know these cells are dependent on EWS fly. If you knock it down in low throughput, it's very clear. But we just got back CRISPR data literally in the last days, and fly does score as the top dependency compared to other cancer cell lines that were screened. And the one line that you see in the middle that does not score, that line actually has an ERG fusion, not a fly fusion. So I think the data is going to be quite clean. And normal fly is not, wild type fly is not expressed, so just the fusion is expressed. We know that there are a series of recurrent copy number events. We've known about these as well for quite some time. But very recently, there were a series of papers that were reported that looked at the genomic landscape of Ewing sarcoma. This was work that was led by Brian Crompton in my lab and Chip Stewart from the Broad. There was a publication by the St. Jude group in collaboration with Olivier Delatra's group from Paris, and then a publication from the NCI. And the theme across all three was very consistent. These tumors are extremely quiet in terms of their genomic complexity. And the one, I think, most striking finding was that there were recurrent mutations in a gene called STAG2, and I'll return to that again. And that these events tended to occur, there was an association of STAG2 events in TP53, but not 100% overlap. And so in light of these very quiet genomes, a very defining event, EWS fly dependency on that event, we did set out in a smaller pilot screen to identify new potential dependencies in this tumor. And so we did ChIP-seq to look at super enhancer mark genes in Ewing, looking at H3K27 acetyl levels. We remined data that was published by the Singer group that is known as the genomics of drug sensitivity screening data, and then also did a pooled shRNA screen and compared Ewing sarcoma against hundreds of other cancer cell lines. And what we found at the intersection of each of these screens really was Cyclin D1 and CDK4 as scoring, and that Ewing sarcoma seemed to be particularly sensitive to suppression of these two genes. And just shown here in the hockey puck, this is working not very well, you can see it, is Cyclin D1 in terms of genes marked by super enhancers. You also see in the middle figure that Cyclin D1 and CDK4 score is highly depleted in Ewing compared to the other cancer cell lines in the screen. And then Ewing sarcoma and EWS fly were predictors of response to a small molecule inhibitor of CDK4. And then again, very recently, our data from the CRISPR screen also showed that CDK4 is a Ewing dependency when you compare Ewing to other cancer cell lines in a CRISPR screen. So expression of Cyclin D1 and CDK4 in Ewing compared to non-Ewing, they are both extremely highly expressed. This also holds up in primary patient tumor material. So I'm sure you know Cyclin D1 and CDK4/6 are quite important in the cell cycle pathway. And over the last several years, specific inhibitors developed against CDK4 and 6 have been developed. So we have worked closely with Sung-Kyu Kim from Novartis to study LE011, which is a CDK4/6 inhibitor. This molecule, as you would predict in Ewing cells, induces a G1 arrest. But what we think is quite interesting is that this compound also induces cell death in Ewing for reasons that are not yet clear. So in general, the molecule in most cancer cell lines where they're responsive, it induces just an arrest and not death. We also found that the compound was active in vivo, so we looked at a drug dose that had been used most frequently in the literature and then adapted to a dose that we think is the more clinically relevant dose, which is 75 mg per kg. We do see inhibition of target in vivo. We see downregulation of phospho-RB, again with a dose response. We see a delayed progression of tumor and stasis of tumor at the higher dose, as well as prolonged survival with one cycle of treatment. So we're now interested in understanding why is it that Ewing cells die and in identifying more highly effective combinations of drug. We think that you would need, at least in the clinical context, to use this compound in combination with other molecules. And so in that study and in ongoing studies, we're interested in identifying what are biomarkers of response to EWS, or EWS, what targets are potential targets that EWS fly as a biomarker of response to. But we're also now interested in looking at synthetic lethal relationships with another molecular event in Ewing, which is the loss of function of STAG2. So a little bit about STAG2. I think this is one of the more exciting findings in the deep sequencing events of many cancers, that STAG2 and other members of the Cohesin complex are recurrently mutated. And I just show you here a handful of those diseases, including Ewing sarcoma, AML, GBM, and bladder, where there have been focused publications on the role of STAG2 or mutations in STAG2 in these diseases. And I would make the argument that if you were interested in identifying synthetic lethal events with loss of STAG2, it might be easier done in Ewing, where the genomic complexity is much less, than in more complex tumors such as bladder cancer. So mutations in STAG2 occurred in about 15 to 20% of Ewing tumors. These mutations led to loss of protein, as shown in the Western blot across a panel of cell lines, as well as by immunohistochemistry, so we have a very nice marker to look at in the clinical context. We also found a cell line that had a rearrangement involving STAG2 that led to loss of the wild-type protein. So STAG2, as I said, it's a member of the Cohesin complex. This is a 4-subunit complex that includes most commonly SMC3, SMC1A, RAD21, and then either STAG1 or STAG2. And then there are other members that are important in meiosis, but usually are not expressed in normal cells. Otherwise, this entraps DNA, the structure in its ring structure, and it's important in multiple functions. It's quite a complex protein complex. It's important in sister chromatin exchange and chromatin architecture, transcriptional control, as well as in DNA damage and repair. I think interestingly, there are mutations in members of the complex in a developmental syndrome called Cornelia de Lange syndrome, but there are no reported mutations in STAG2 in this syndrome. And these patients, to our knowledge, are not more prone to development of cancer, which I think is also interesting. As I said, the complex is mutated in multiple cancers, but in Ewing, only STAG2 mutations have been observed. So in our study, we found that mutations in STAG2 are actually associated with metastasis in patients at diagnosis. And when we studied the transcriptional profiles of these tumors when they had expression of STAG2 versus when they did not, the tumors with a lack of STAG2 were enriched for metastasis signatures. So one angle that we're quite interested in exploring now is whether we can functionally show that loss of STAG2 is relevant in metastasis, and those studies are ongoing. The St. Jude Paris group showed that these mutations are also associated with poor prognosis, particularly when we have the co-occurrence of P53 and STAG2. So we're now engaged in a much larger study with Children's Oncology Group to evaluate over 500 Ewing sarcoma samples where those patients were all systematically treated on the same protocol to ask, does this finding hold up in a larger study? And then we'll also separately look at a cohort of patients with metastasis to see if there's a higher incidence, and again, in much larger numbers. So these are the two slides I'm going to talk about tumor heterogeneity. There's actually very little data thus far on tumor heterogeneity in pediatric cancers. These cancers, as we said, are rare. When we do biopsies, we get very small amounts of tissue, so they're small, usually radiology-guided biopsies, so there isn't that much tissue available. And so the first generation of sequencing papers are just emerging, and we really now are just catching up on looking more closely at heterogeneity. But it's clear that it's there. I think these are some very nice examples in the next two slides. This shows from the St. Jude work, cases of patients at diagnosis versus relapse. And when you look closely by immunohistochemistry, you can see some variability in STAG2 staining at time 1. And then at relapse, there's marked enrichment for loss of STAG2. I think, again, suggesting that this is likely going to hold up as a poor prognostic marker. And I will also say to date, we have no markers of outcome. The way we divide patients is you have metastasis overtly or you don't. So it's still pretty primitive in terms of how we classify and treat these patients. So I think any biomarker will be an advance for us. So in our own work, we found that there was a patient who at relapse 1 had a P53 STAG2 mutation, then at relapse 2 had completely different mutational events in STAG2 and T53, again suggesting that there is certainly some clonal heterogeneity within these tumors. And then we also had a very interesting baby who had unique pattern of mutation at time 1 and time 2, and the only unifying lesion was the EWS fly fusion. So there was work initially that was published in Science that's suggesting, at least in some cancers, events in this complex lead to aneuploidy. And this does not seem to be the case in Ewing sarcoma, nor in acute myeloid leukemia. So in our data, and in also data of others, we've looked at whether there was a difference in tumor ploidy with or without STAG2 expression, and there isn't. And we looked at the fraction of the genome affected by copy number alterations and corrected for P53, known to lead to copy number alterations when mutated. We didn't see a difference. So we think in Ewing, this is likely to be a chromatin and transcriptional effect rather than an effect on inducing aneuploidy. But I think that if there's a subtle defect in sister chromatin exchange, you could imagine that this might engender a synthetic lethal dependency. And this is really what we're actively exploring in the laboratory. So when we knock out STAG2 and Ewing sarcoma cell lines that express STAG2, the cells grow fine. They don't grow better, they don't grow worse than plastic. And so while I think this makes looking at a phenotype more complex, and we're now exploring metastatic phenotypes, it does allow you to do screening. And so we're now engaged in a screen to compare cell lines that express or do not express STAG2 and whether there are unique dependencies in the absence of STAG2, either with CRISPR screening or small molecule screening. And then we've generated a series of isogenic lines with and without STAG2 knockout, and we're now actively as well screening and expect to have data pretty soon. So we're excited about the prospects here. And I think this is a case where if we find something interesting, these are children that we think are higher risk. We really have no interesting therapies in the lineup for these children. And we think that these relationships likely would hold up in other cancers where STAG2 is lost. And in AML and myelodysplastic syndrome, there's some data suggesting that these events may also be a poor prognostic feature. So with that, I'd just like to make some concluding comments. Pediatric cancer genomes are frequently simple. They're core defining genomic or epigenetic events. I think we can learn a lot from studying these. In some pediatric cancers, increasing genomic complexity is really associated with worse outcome. JMML is one unpublished example of this. However, some very genomically quiet cancers are still highly resistant to cytotoxic chemotherapy. And I think it's interesting that at least many of these are defined by key events in epigenetic factors, such as rhabdoid tumors and MLL rearranged leukemia. And I do really think we can learn a lot with genomic simplicity and really leveraging it as we've done and will continue to do to find new biomarkers of response to drugs and new synthetic lethal relationships that we can then extrapolate this information in the study of other more heterogeneous complex tumors. So with that, I just want to thank the laboratory. I tried to highlight project leaders along the way, particularly Brian Crompton, who led the Ewing sequencing work, who's also now leading our STAG2 efforts, and Li-Ying Chen, Alyssa Kennedy, and Monica Valaparelli have led the CDK4/6 studies. And then Mignon Lo's lab is working with us on the JMML work, and also our many collaborators. Thank you. Happy to take questions after.